Hello, everybody. Today I'm going to present you our SureTech PCR system for detection of pathogen and other spoilers like Brettanomyces. So I will start to explain you our workflow for Campylobacter, and we will really see all steps from the enrichment to the confirmation. So we will start with this Diloflux Pro, which enables, in fact, to prepare in a simply way your sample. You will just have to put the bag here, so you have some kind of magnet that open it for you. You can select different mode, but here I just selected a mode to have a dilution, a 1 to 10 dilution factor. This is what we need for Campylobacter detection enrichment step. And so here we have different protocol depending what you want to test for Campylobacter, depending if this is raw meat, this is processed meat, etc. So in our instruction for use, you will have, in any case, all the information. So if we look this system, you just need to put the bag, to operate the tear, and after to add your sample. Here I will take. So normally, with the good practice, everybody is wearing gloves, and specifically for PCR testing. Sorry. Exactly. Yeah. Match the outfit. Exactly. So I will add here my sample. I have my 25 gram 0.05. Okay, we're not in real condition. Let's see that it's okay. And so here I will start just to press go, and the Diloflux will add automatically the needed amount of media for the enrichment step. Once we have finished, we are going to use here the homogenizer. Okay, so we just take the bag, we open the device, you see how it is. So you have the big pail that will scratch the sample. So you place the sample, you just go, and by pushing, it starts automatically. You just need to set up the speed and the time. I just put a short time here for the demo. Yes, it says that it's running, in fact. And when it's finished, normally, it's come back to green. So once you see it, I've been quite efficient in 15 seconds. And now we're going to proceed to the incubation. So imagine that we have an incubator, we close the bag, and we put it for Campylobacter. We will go at 42 degrees for 22 hours minimum and up to 30 hours. Then, when you recover your enriched sample, we arrive to the preparation of our PCR method, our SureTech workflow. And what we want to do first is to have our DNA to be detected. So from this enriched sample, we are going first to do the lysis step. For this, you will need to aliquot a bit. So you will just take, imagine that you have much more devices and things. So I will go here. I use a filter bag so I can go in the filter area to avoid to take some particles. I will put it into this aliquot tube. And from this, I will transfer into some lysis. So imagine that we had collected all our sample of the day, etc. Okay? And now we are going to use our shooter kit. Everything is inside. In fact, you have already the lysis tube, which contain the lysis reagent. You will have the PCR tubes, which contain the lyophilized PCR mixed. And you will have as well the proteinase K. Okay? So with this proteinase K, this is what we will need to add first into our PCR tubes. So this is our lysis. Sorry, I made a mistake. It was the lysis tube. So this is the lysis tube. You see here that we have already pre-filled reagent inside. Just what you need to be sure is that the liquid is really equal. Sometimes some drops can be stuck on the top. So you just flip like this, and after you can open. So here, I suppose that I have 8 samples. I take a full strip, but imagine that you need to only have 4 samples. You can cut the line. So I'm going to show you. I just put this here. Okay. You see, this is the lysis. And we have some cutting tools here. So imagine you just need 4. You will place your in the middle. If you need just 1, well, you can even take 1. This is really simple. You place it where you need. And after, you just press, and it cuts into equal parts. Very simple. Okay. So now we have our lysis tube, which are opened, and we will need to add 10 microliter of proteinase K. You can use, I don't have this tool today, but for sure, a multi-stepper, a stepper pipette to allow you to take a big amount of the proteinase K and to just after distribute in repetition the proteinase K in the lysis tube. So you take the proteinase K, and let's go for 10 microliter. Here, for reason of plastic save, I won't change the tips in between each things, but normally we really recommend if you just use a single pipette to change your tips at each distribution. So let's go. Oops. So with the stepper pipette, it's for sure quicker. Okay. I'm down with the proteinase K. And once the proteinase K is added, we will just need to add our sample. So we will transfer, in fact, from this sample tube here, once again, we will transfer our lysis, and let me just check the volume, 10 microliter as well. So once again, for each sample, you will take a new tip in between. Okay. Then Once this is done, what we need now is to seal. In fact, we have two options to seal the wells here. We have some doom cap. It was already in our former kits, but I would say that now I would really recommend to not use anymore the doom cap, but to just use those adhesive seals that allows after to transfer without opening again the tube, because here we will have the DNA, and so to avoid any spread and any contamination in the lab, we really recommend to use a perceivable film. So you can definitely once again select and take only what you need. Everything is ready to be cut. And so you will just need to apply it on the strip. Okay, once this is placed, you have this tool to really apply strongly to be sure that it is fully stick. Okay. Once this is done, we just need to insert. So we take a special tray for the Simply Amp, and we will put the wells inside and go to the Simply Amp, placing, and we will just need to push to make the run. So we have prepared already some methods. So you just need to open your method. For example, here I have my SureTech method. Okay. And you see here the cycle. So we have 10 minutes at 37 degrees, followed by 5 minutes at 95. Here, it should destroy the bacteria membrane and free the DNA. And then we will have a cool down at 10 degrees between 2 minutes. And after, this is stored at 4 degrees. What it means? It means that, for example, you have finished your lysate, but you need to do something and you want to run your PCR in a second time, you can. So you can leave here. This is stored at 4 degrees. And so it gives you more flexibility in your lab organization for any reason that you would need. Once, so you launch the run. So here we have all in all a run of 20 minutes, let's say. And we will recover our lysis tube with our extracted DNA, okay? So here we are going to place on another holder that for the PCR tube, you see here the PCR tube. I will insert my PCR tube here. I place again my lysis tube here. And so I don't need to open. And what I need to do is just to transfer my lysate to my PCR tube. So as I mentioned, you have some dehydrated pellet containing. Always be sure that the pellets get a kind of white-yellow color and are really, not look like with another color, things like this. You should not use it. Should have perhaps something, but this is really the typical aspect of the pellet. And so we need to rehydrate them with the lysate, and we will transfer 20 microliters. So here, what we can do is to use definitely a multi-channel pipette that will save us time. Okay. And so here you just need to come and to pierce the film. Come on. Yes, enter. Everything is good. Oh, I forgot to open my strip. Sorry. I will just, it's not, yes, thank you. It's not good practice, we agree, but I'm not in a real lab, but I hope that it gives you the real flavor of what we are doing. Thank you so much. And so we are going to insert and to dispense the 20 microliters. And we're going to close the PCR tubes with optical caps, okay? And once you put the optical caps, you will never open those tubes again. You will trust them like this, because you can imagine as well the potential cross-contamination that can occur by opening those tubes. Okay, so you see that it is not well, mixed. So what we need to do here is to vortex those strips for a while. Okay. If it's not enough, you really need to check. When you work with full plates as well, you can use, the vortex with the 4 places to put the plate directly. Works very well as well. So here we go. This is completely dissolved. We are good. And now we can just go to the QuantStudio 5 thermal cycler. So for sure, at the beginning of your test, you will have set up your software that drives our QuantStudio 5. This is really simple to use because, in fact, you have a list of all our SureTech kits. You select the kit, you select the number of samples. This is very intuitive, and you will obtain very simple analysis report. You will have color code. When the sample is positive, it will be red. When it's negative, it will be green. And if there is any doubt on one well, you will have an alert, an orange color on the well. I want to precise as well that in each PCR tube, you have an internal positive control. So when you have a negative, you are sure this is a negative result and not an inhibition of the PCR reaction. So this is included in all our SureTech test kits. So now you have set up your sample name, you have selected your, for example, Campylobacter or Salmonella or Listeria monocytogenes kit, because the advantage as well is because of a unique thermal profile for the lysis step, you can mix in one plate, in one run, different parameters. So you can run on the same plate Salmonella and Campylobacter. That makes sense for poultry, for sure, meat samples. So once you have set up everything, you just need after to go to the QuantStudio 5. That will be fully driven by the computer, and you just need to insert your plate and to launch the run. It will take an hour and 10 minutes, something like this, to obtain your full result. So if we look at a little bit, so let me just take up this glove, and perhaps you will see it on the screen. Oh, sorry, it's not. Okay. So this is how the result will be presented in the report. So you see here, you have the run, the result. So you see all the wells will be given. You can select it here, all the thing that you need in your report. So this is really simple. You don't need to, you know, everything can be connected to the LIMS. You don't need to export or things like this. It can be already exported directly to your network in some specific files. All the system can be connected to your lab network. So you see that there is really 3 main steps to prepare the sample, to execute the lysis, and to run the PCR. And this is each time for all our SureTech kit, the same procedure, the same protocol. Where you can find some differences, it's in between gram-positive and gram-negative. Sometimes you will need to add one other reagent to the lysis tube. Or, for example, and now I'm going to talk to you about another specific kit that we have developed for one big beverage company. And I think we can name it because we have made a testimonial with them recently. So with Coca-Cola, we have developed a kit for the detection of Brettanomyces cerevisiae and Brettanomyces and Saccharomyces cerevisiae. Okay, so with those this kit, what was the goal? In fact, in the beverage industry, for testing this type of parameter, it was a very long procedure, up to 7 days. Up even for the confirmation, etc., 8-10 days. So we decided to work on this problematic, and the solution was to get out from, let's say, the traditional and conventional method, and to go with a PCR kit to really shorter and the enrichment step and the analytical step. So all in all, we come with a solution with a 3-day of enrichment, and after the PCR in 2 hours, let's say, to 3 hours. So we really reduce from 7 to 4 days the analysis. What is the difference, for example, for this type of kit? So it can be applied for all environmental samples, but as well on end products. And we need to here add a little bit of steps, but that really allows to reduce the analytical time. So in the kit, you will have, like for the other SureTech kit, the lysis tube. You will have the PCR tube. You can see as well that it is a different color of the plate. So in the lab, you cannot make mistakes. You will have for each parameter a different color code for the plate. So salmonella is red. Here, so for the bread, it's green. Listeria, it's blue, if I don't make mistake, etc., etc. And we have here 2 other reagents. We have the lysis reagent 3 and the lysis reagent 3 and dehydrated one. So here we need to reconstitute this one. So what is the workflow of the SureTech kit? Here, let me just clean a bit and put what I don't need anymore. Okay. So for this workflow, you see that once again, if you want to run a full plate, you can run the full plate. If you need to cut, once again, you can use the cutting tool, etc. But what is, I think, important, it's to really realize that here you will have, once again, few steps in comparison to what is used to do. So you remove the seal of the lysis tube. Let's take one. Okay. And now we will add, so imagine that we have already prepared the reconstitute this little powder here, and we will add 10 microliter of this region. So you just imagine that I had it. Then after I will need to cover the well with a first plastic film, and I will add in a second time my lysis, from my PCR tube. So I will add my sample. I take the sample from the enrichment. I had the sample. I put another film, and I had the lysis, the third lysis buffer. So once you add all those, you will have 2 layers of films. You go to the SimplyAmp and you run your extraction for the DNA. You recover your strip, and once again, you will transfer to the PCR tube and goes to the QuantStudio 5 device. But once again, I would say that perhaps you will need to here reconstitute this region and add the 2 regions to the well with the sample. But in comparison to what the beverage company used to do for this parameter, they save a lot of time and a lot as well of technician time, technician handling. Okay. Do you have any question about the system, about the workflow? I forgot one thing, the confirmation. Once you arrive to positive result with the QuantStudio 5, so as I mentioned, we are able to provide you the workflow from enrichment to confirmation. And so for the confirmation, if we come back to Campylobacter, you will just need to strip from the enrichment bag a Brilliance, Campi agar, or something like this. You will incubate for 2 days, I think, for Campyloaf, and you will recover your characteristic colonies. And then either you follow the ISO colony confirmation, etc. Either you can use our obese Campylobacter biochemical galleries that will help you in identifying the strain and confirming the strain. So this is very simple, and we can really provide you all the Brilliance specific kit to obtain the characteristic colonies for the confirmation. So this is, we have for Brilliance for Campylobacter, we have Brilliance for E. coli, we have Brilliance Salmonella, etc. If you are as well a user already of chromogenic media, of Brilliance media, as an alternative method for your screening, the SureTech can be used as a confirmation. I want to as well mention that we have some QC, quality control organisms. So you need to be sure that the enrichment media that you use is efficient. You need to be sure that the Brilliance media to select your characteristic colony is performant as well, that there is no other microorganism that could grow on it, etc. So you need to run on all the batch that you receive this quality control. So we have calibrated loops. In fact, you can see it with the microorganism that you just need to spread. This is fake, this is for demo. And so you definitely will save a lot of time by using those microorganisms, and be sure of the quality. This is a TCC strain, so you can be sure once again about the quality here of your control, of your quality control. Okay, I think I give you all the information from my side, but perhaps you have some more question, Jarek?
Tak jo, pokud máte otázky, ptejte se, pište do chatu, klidně je formulujte česky a já to budu překládat. Prosím, pojďte na to. Sandra, so you mentioned the whole workflow, so maybe it would be useful, like, to summarize the
Yes.
individual steps, including the real time you need, like, for every steps that we
Okay.
could demonstrate. I mean, how much time, I mean, is needed to spend
So-
with every single step. Yeah, and then all together.
Yes. So for, let's say, pathogen, and I don't talk about the Brettanomyces kit here, but for all Campylobacter, Salmonella, Listeria, etc. In general, we are in between for the time to result global as at day +1. So in 24 hours, you can obtain your result for Salmonella, for Listeria. Depending after about the enrichment time, it can be up to 2 days, etc., depending your parameter. But this is very faster than the ISO traditional method, okay? For the sample preparation, you saw that it can be very quick to be prepared. In addition, we are providing some quick bag, feed bag. So you have bag with the ready-to-use media, and you just need to plug the tube here to the bag, and this is ready to use. If you prefer to have bags with deshydrated media, it exists as well, and you just need to press the water into the powder part, and it mixes it automatically. So you have both solution. The second one is sometimes better for saving space, storage space in the lab, because this is a bit less, less volume, let's say. So after, for the homogenization, it took in general 5 to 10 minutes, in depending the, so let's say for the sample preparation, for sure it needs to be consider to your number of sample, but for one sample, let's say that you will take 1 minute, less than 1 minute to prepare it. Homogenization 5. Then after the lysis step, from a pure handling part, you will just need to transfer sample into the lysis tube. So let's say in 1 or 2 minutes it's down. Here you have the cycle of the lysis, which is in between 20 minutes, not more than 20 minutes, let's say 15. And then after, you have the PCR run of 2 hours. Though the biggest part, in fact, occurs at the enrichment step. This is where you spend the most time in the lab. But this is something which is mandatory. We don't know how to do microbiology without this part until now. So yes, and so all in all, you have very a 2.5 hour PCR run, but not more.
Thank you, Sandra. Just question, maybe it's a question from somebody who sees it for the first time. I've seen you've been pipetting a solution that had a blue color, so
Yeah.
it has some intention that it's blue?
Yes, exactly. So you see that our lysis tubes contain a transparent color. When you need to add the proteinase K, this is a kind of just color control check. Oh yes, I had it, my proteinase K in the well. No doubt about it. So this is really to be sure, visually speaking, that you had the proteinase K. And once again, for the color, we push it, as I mentioned, for the PCR plate. So you cannot mix, you cannot take a salmo kit. If you use to it, you will use to the red color. And if by mistake you take the another kit and you start to say, oh, it's not the right one. So you have an additional check with the color of the plate. This is why our, let's say, sentence is easy to be sure, but it is visually secured.
Yeah, thank you. One more thing, could you summarize all pathogens that can be detected by this workflow, please?
So with the SureTech solution, so as I mentioned, so we have a very complete and very nice portfolio for Salmonella testing. So we will go for Salmonella species, but we will have as well a multiplex for Salmonella, for targeting in the same run Salmonella, SPP, Typhimurium, and Enteritidis. We have as well a Salmonella kit for quantification. So this is something that now it's a need for all the US market, but with the global supply chain and the exchanges in between the countries is something that can be considered. We have, so for Salmonella, I think that I mentioned everything. In addition, so we have Listeria SPP, Listeria monocytogenes. For E. coli, we have different kit that allows you to target specifically O157H7, but you can as well go for steak and EIE. So you have different kit for E. coli depending how you want to target and to screen, I would say. We have Campylobacter kit. We have, I forgot some.
It's a test for you, so
Please help me, yes.
I'm not the right one to help you, so yeah.
Let me just, I can tell you just with a brochure, I can go to the brochure shortly. Listeria, E. coli, Staph aureus, the bread for the spoilers. And as well, what I can mention, so here we are talking about pathogen, but this QuantStudio 5 can be a platform as well for testing much more than pathogen. We have as well a solution and kit for GMO testing, for animal species, for, yes, different halal, for example, testing, vegan testing. So this is an open platform that allows you, in addition to, for sure, run all our SureTech kit, but as well, if you need, for example, to make some R&D or to develop some homemade kit for any reason, you have specific channel dies in the QuantStudio 5 that you can calibrate for your own purposes, etc., etc. So the device itself, it's an open platform for pathogen testing with the SureTech workflow in terms of ease of use. You won't find on the market something such easy to use in between and with all the control that we included to avoid any, like the pierceable film for avoiding the cross-contamination when you open the cap, etc., after the lysis step. So this is very efficient. And in addition, you can run all other PCR analysis that you wish.
Yeah, I don't know if you mentioned Legionella, but this is something what's included as well.
Legionella testing as well. We have a full solution for Legionella. We have as well launched recently a kit for a new Legionella strain targeting. I forgot exactly the name of the strain, but it's something that in Europe is raised. And so we have developed, in addition, this multiplex for being able to integrate in the screening this specific strain.
Yeah, thank you. Napadají vás nějaké dotazy ještě, prosím? Okay.
I hope it was clear.
Yeah.
Thank you very much for your attention.
Yeah. So it was clear.